Ok. Buenos días a todos. Vamos a empezar el último día de sesión del curso. Qué bueno que la mayoría, creo que casi todos han estado viniendo. Me da mucho gusto. So,
Great. So today, day 5, we're going to talk about genetics as tools for biotechnology, but we decided originally we're going to do our tools, Stanley, me, and then switch and give you something else. We decided I'll just give you my tools, and then I want to give you a talk about a genetic journey, and then Stanley will take over. Okay? Okay. So I hope you can see this. Let's turn on the movie. I have a colleague who, as a postdoc a long time ago, he discovered DNA ligase. Okay? Pretty well known for that discovery. And he worked on DNA polymerase, and he was Filipino. And after all, he was at Caltech, Stanford, and then he went back to the Philippines, and of course, there's nothing. You can't do molecular biology in the Philippines, right? But there was a discovery by a guy named Alan Cohen at the University of Washington about these marine snails that you'll see that eat fish. And so the Philippines was full of these marine snails, and so he would purify their venoms and inject them into mice. Like put the mice in a cage and ask how long does it take before these kinds of things. But when he managed to get a job back in the United States, and he started doing molecular biology with these toxins. And so these CONUS species, these are marine snails, and they eat fish, worms, and other snails. Okay, so they're predators. And so how does a snail catch a fish? And catches a fish by injecting toxins. And there's 1,000 of these species. And each one has a venom of about 200 different toxins. Okay. So he was hoping to figure out a way to try to, he's purified many of them. Actually, one of these toxins is used as an anti-pain drug for people that are dying of cancer and they become immune to morphine. And so they're in horrible pain, but you can inject one of these toxins through a spinal tap and it relieves all their pain. So they can be very important medicines. And there's others that are related to insulin, and all kinds of stuff. But how does that get to us? Okay, so I'm just going to show you a brief little movie. You can see that. You want to turn the lights down a little bit? Senor? Yeah.
The smaller ones on either side are its eye stalks, and in the middle, a deadly proboscis.
Ooh. Like alien.
A harpoon loaded with the neurotoxic mix so complex there is no antivenom to thwart it. It's armed with at least 100 different toxins, more than any other creature.
Immediately the stomach comes out. It's paralyzed.
They like to burrow into the sand and mate.
So there's different ways of hunting strategies.
The cone snail fires its harpoon.
He's underneath the sand now.
Poisons free. once
It
it's
comes
ready,
out.
the cones
Oh.
are
Dinner.
Dinner. Oh,
The snail is no longer hungry.
pretty cool, huh? Okay, we can turn the lights up a little.
¿Prenderías la luz? Gracias. Pásenle su casa.
All right. All right. So, like I said, there's a lot of each venom has like 200 toxins, and some of these snails will kill people. So they've been known to kill people. They'll harvest these snails because they're beautiful shells, and they'll put them in their bags and diagnose them. They feel a sting, and by the time they get to the hospital, they're dead. Or they'll be having a snail that is covered with some mucus, and they'll scratch it with a knife, and then they're dead. So the toxic ones, for humans, one venom is enough to kill 700 humans. This is a very powerful venom. And so he's been studying that because, as you can see, if you're a snail and you want to catch a fish, you've got to paralyze everything. So it hits every sodium channel, calcium channel, every neural channel, every synapse, of all these different toxins, right? And so they're actually used to study neuroreceptors in humans. And so he asked us if we can help them try to purify these things easier. And so we went to our flagellar system. And
Before you go,
yeah.
can you tell me about the golden fleece and one thing?
Oh,
Quickly, because it's a cool stuff.
that didn't work out very well. Yeah, so a couple years ago, Toto, back in the 80s, there was a conservative senator in the U.S. government. He would put out what he called the Golden Fleece Award. And that was to pick on scientists that were doing what he thought was a waste of taxpayer money, right? And then another senator countered that with what he called the Golden Goose Award. So we know that it's the goose that lays the golden egg. And the Golden Goose Award is when you fund research and you have no idea where it's going to lead you, but it leads you to something incredibly important. And so he got the Golden Goose Award for his characterization of these marine snail toxins. So why would you study marine snails? Why should the NIH National pay you money to do that? Well, like I say, now there's drugs that are used for treating humans. So you never know, right? Okay, so remember our flagellar system? So it's a little fuzzy here, but I hope you remember. We build this hook basal body. There's this anti-sigma.
Esa palabra de la
See it on that one. Maybe I should draw that. No, that's okay. So there's a sigma factor, an anti-sigma, and then once this is complete, there's a switch, and the anti-sigma, this flag goes out, and releases the sigma to turn on the late gene. So we can engineer our salmonella to get rid of all of the late substrates, the fly seed, the hook filament junctions, and have only one protein that's being secreted, and that's our flag M, our anti-sigma that goes out. And here we are. Okay, so this is cellular, secreted. I'm going to have to look here because I can't see
Se
something.
vea. Si puedes
Okay.
apagarlo para que se vea.
Here's actually our wild type flag M. It's mostly secreted. Okay? If it can't go out, you see it's not secreted. Actually, it's an auto repressor, so it'll shut itself off. And then if it's deleted, of course, there's nothing there. And then, what do we have? Oh, we have it under, here we have it under Ravenose control, so you can turn it on with the Ravenose. And this represents 3 different fusions, where we have, at the end of flag M, so we have the hook phase body. We can fuse that to a conotoxin. And now, since this is the only protein going out, it'll take whatever's with it outside the cell. Okay. So these are 3 different conotoxins from photo, and as you can see, they're secreted. And so the idea is it's a one-step purification of protein. You just grow up the culture, you spin down the cells, and the only protein left is whatever you fused to flag out. Okay, and that's what this paper was about. So it just shows some different fusions. This is one that inhibits a sodium channel, and you can see this is a normal sodium channel reaction. And if we had our purified M conotoxin that inhibits that sodium channel, there you see it works. So we can get out active purified proteins. And so I had a student by the name of Dan Lee. He was a little sloppy in the lab, but he had a good business sense. So he thought, I can start a company, right? And so he decided to start a company where he used this to purify proteins. And he raised $1 million, started his company, and we got a terrible business officer. The guy was a complete idiot. I said don't hire him, but Dan still hired him. And the guy just convinced Dan that we needed to purify insulin, right? Oh yeah, it's a $4 billion industry. Let's make insulin. So one of the things that Dan did was he was working with these big fermenters where you can make 400 liters of cells. You can pump in oxygen. And normally, if you look at growth, how do I want to present this? So cells grow, and then they stop at about an OD of 2. Right? They hit saturation and they stop. If you work with these fermenters, you can get them to go to ODs of like 40. So super thick concentrations of cells. So that was a brilliant idea. Unfortunately, Dan didn't read the literature from our own research in the lab. So remember that class 1 operon, FLHDC, that turns on everything, the master operon? Well, if you look at its expression, it goes up and then goes down about OD1. So yes, he can grow up a lot of cells, but guess what? There is no protein. Very smart. Anyway, so after wasting all the money, fortunately he was smart enough to sell the technology to a Norwegian company. And now those guys know what they're doing, and so they're using this to make protein. So that's how we can use a simple flagellar secretion system, one-step purification proteins. So that's my biotechnology spiel. Any questions? And actually, I could point out another thing. There's one on here. So John Collier at Harvard, I was talking to him. Where is it? Down here. Right. It is. Oh, here it is. Right. So we were able to purify some proteins that were very unstable in the cell, right? That normally people can't purify. You express them, they're degraded. But if they're expressed and secreted, there's no proteases to degrade. That's another thing. Okay. Yes. Oh, there you are.
How big can the protein secrete be?
So the answer is we don't know, but we do know that the flagellum, which is secreted, is 550 amino acids. So we know we can secrete large proteins. There are some proteins that can be tightly folded that get stuck. that's one limiting factor. You have to figure out ways around that. Good question. Any other questions? Yes.
What if we want to measure the amount that the protein we get? It is possible to control that part, how many protein we get, or we need another method for that?
How do you do a Bradford assay or something like that? Is that I don't. You just want to measure your protein levels that come out.
I think the amount that the protein that we obtain for this, maybe we want to produce a lot of protein.
Yeah.
And how can I measure how many protein I get? And
Maybe
I control
this
that
question
part.
is related. How do we know how many units, for example,
Yeah.
lyc, FLGM?
Well, you can purify the protein and just measure how much protein is there.
And I
That's
think
pretty standard biochemistry.
Just by regular
Bradford assay.
We purify the protein sometimes.
Huh?
When you purify the protein, you need how do you distinguish? Because Bradford doesn't distinguish if you have lyc or.
No,
That's
so-
the question.
Only one protein is going out. We engineer the cells that nothing else is going out.
Okay.
I mean, you could still run this on protein purification columns and just show that there's nothing else there. We've done that. That's all you get. All right. Any other? Okay. So that's our biotech. Now that's it. Norway. Toto, so obviously we can engineer because we have this, we have under at the air locus, we have flag M. That's dark. Sorry. It's all right, black works. We have flag M, and then right after flag M we have a tetra. And then we can bring in any conotoxin, and do a TET-sensitive cross to replace the tetra and get our M fusion, right? So it's easy, one-step insertion, turn on the Ravenous, and that's it. Okay. So the question is, can we figure out how to set up a robotic system to hold 1,000 species times 200 is 200,000 toxins. We haven't done that. Maybe if someone gives us some money to set up a company, we can do that. But that was when it works. That's our venture into biotech. Okay. Now,
Black button straight in front of me. Oh, I can do that.
let's see if this is the. No, it's the.
It's a projector.
It's a projector. Okay, well, we'll do the best we can. So I thought I'd take you on a journey. So we spent the week talking about genetics. And so I wanted to take you on a genetic journey from flagellum gene expression to mRNA translation. Maybe we can turn some lights on in the back. Maybe.
Maybe you can put it as presentation and it will be look bigger at least and we'll get better.
Yeah, let me get started. All right.
There we go. Yeah.
Better? So this is work done by my research professor and my wife. And this is our grad student that did this, Madison. And this is Venki Ramakrishnan, who got the Nobel Prize for the ribosome structure. He did a critical experiment that helped us figure this out. Okay? So I thought, I just want you guys to appreciate. Remember we talked about the whole you can't always get what you want, right? But genetics is not about necessarily, someone asks, well, what do I do with it when we're talking collaborative, but I don't know, sometimes you just have to think and get results and follow them, right? It's the stuff that you don't expect that's going to take you into new discoveries, right? Okay. So this is what we work on in my lab. Can you see them? Aren't they beautiful? Swimming salmonella, right? When they rotate these flagella at speeds of like 300 cycles per second. Vibrio ocean bacteria will rotate them at greater than 2,000 cycles per second. So imagine 1 second, 2,000 times. Amazing. That's why biophysicists love these things. Okay. And this is what we figured out over the years. We have a master operon. It builds first a type 3 secretion apparatus, and then we start building parts. There's a dry shaft that gets to the outer membrane. There's some bushings around the dry shaft that punch a hole, and then we grow the hook. And I didn't talk about it, but there's a ruler that actually measures how long the hook is. And when the hook is long enough, the bottom of the ruler flips the switch. It says, all right, no more rod hooks. We're done. Now we can secrete filament. And that's when flag M has a filament type secretion signal, and it goes out of the cell to release the sigma factor, and now you can make filament. So that's how we couple assembly to gene regulation. Yes?
Is there a similar rule to measure the length of the flagellum itself?
No. It's hindered diffusion. So if you, at the start, when they're growing filaments, they are secreted at rates of like 20,000 amino acids a second. So because it's such a very fast secretion rate, it was really thought to be coupled to ATP hydrolysis. Bob McNair was pushing that. But that wouldn't work. So in the sex system, so the system that sends these proteins out that make the bushings, that's ATPase dependent, and they go out at rates of 10 amino acids per second. Right? So this has to be directly coupled to the proton motive force, and it is. So protons flow in. So protons actually also are used to turn this, but also to secrete the protein. It's the proton motive force.
And related to the speed, is it known why some are much faster than the other?
If I delete,
I mean, in the marine bacteria and salmonella compared to salmonella.
yeah, it's well, they also use a sodium channel using the sodium gradient to drive speed. If I delete ATP synthase, so cells will still make ATP from glucose, so substrate level phosphorylation. And they make small colonies, but their proton motive force is so great now because they're not using the proton motive force to make ATP. They swim like crazy. They go from 300 to 1,000 cycles per second. So that's what's limited.
Thank you.
So, what's the equivalent in Spanish?
¿Todo bien?
¿Todo bien?
¿Está bien?
¿Está bien?
¿Está bien? Okay. Estaba is easier for me.
Yeah, just one word.
Just one word. Okay. You guys know what this is?
Yeah.
Transcription to RNA, translation to protein. Okay, this is a movie from Vinke. You can see elongation factor bringing in tRNAs, and they're making protein in the ribosome, and the mRNA is passing through, right? Cute little movie. You know this now by heart, I hope? Right?
Sure.
Yes?
Yes.
All right, and then we've gone over this. DC brings RNA polymerase, lipase body genes, sigma factor, anti-sigma factor. That sigma factor turns on the class 3 promoters. When the hook basal body is built, M goes out, sigma 28 is free to turn on filament. You don't build the propeller until you've made the motor. Let's do some genetics. You can make a fusion. Here's a fusion to chloramphenicol acetyltransferase. You build the motor, and now you become photoresistant. Okay? You break the motor, M never goes out. It always binds sigma. It's inactive. Cells don't express chloramphenicol. What is that? For a geneticist, what does that mean? A positive selection. Yeah. You guys are getting the hang of this. Yay. Yes, we can select for chloramphenicol resistance. This is what we talked about. What do we get? We get null mutants in flag M. If it's not there, the sigma's free. Okay? We get point mutants in the sigma. It no longer binds flag M. So even if flag M is there, he doesn't bind it, you can turn it on. Yeah. We talked about that. Once upon a time, I was just curious. I wonder if there's, I had no hypothesis. Not at all. I was just curious. Are there any essential genes involved in this process, right? So I did a big experiment, 200 independent platings, and I looked for cells that were chloramphenicol resistant at 30 degrees, and then I screened for those that were dead at 42, right? So I could mutate an essential gene that was a little defective and then dead, depending on the temperature. Yes? I looked really hard. I got one, right? And the beauty of genetics, you never have to repeat the experiment. Once you got the mutant, you don't have to get it again. They need to serve. I did have, sometimes I remember you get a rotation student, she'd come in and she'd find this great mutant, and she'd finish her rotation, and I said, well, where is it? She said, oh, I threw it away. 3 months. Oh, there it is. There's the mutant. It's in CRT. So this actually sat for 6 years because I got it before we had the genome of salmonella. So I just stuck it in the freezer. And then after 6 years, oh God, I remember that experiment I was working on 6 years ago. Now we can find out what it is because we have a salmonella genome. And so we managed to get linked transposons, and using those linked transposons and co-transduction frequencies by P22, we could sequence the region where it should be, and that's where it was. It was an essential tRNA called serine T. Guess what codon serine T recognizes? Serine codons. Good answer. Okay. All right. I was not the first to discover this. Okies. I'm always competing with Japanese guys. He was looking for cell division genes. Anything new? And of course, this is an essential tRNA. He got the exact same mutation as G to A. We call it div for cell division. And of course, if they don't have an essential tRNA, they can't do anything at 42. Right? Anyway, so he already had isolated. All right. Now, I'm going to go over the genetic code. Remember this? You've seen this before? First codon, second base, third base. I'm going to show you tRNAs that look like this. So here's the pro-M tRNA, and by having these balls connected by dots, that means that he reads all 4 chromin codons. Okay? Here's pro-L, that reads only these 2, and then pro-K. How do we know that pro-M reads all 4 codons? This is Glenn Bjork at Umeå University in Sweden. Well, he just deleted these 2 tRNAs, and this is the only one left. And they're alive. No? Okay. And you can't read this, but this just shows you some base modifications in the anticodon. The base modification CM05U34 in the recognizes the 3rd codon. That modification is what allows you to see all 4. This is a G at that position, so G can recognize your C, and this is a C that can only recognize G. Okay? There's the whole codon chart. Here's CRT. CRT also has that CM05 modification. And it is the only tRNA that reads this codon, UCA. That's why it's an essential tRNA. And again, we know it can read all four because I can delete these two tRNAs and this tRNA, and they're alive. It's alive. All right, guess what? I looked in flag M. Amino acid 7, voila. There is that TCA or UCA in the RNA, serine, and at 30 degrees, this is defective, and now flag M translation is actually down. That's why we got our effect. How do we know? We can replace that with a different serine tRNA that we know is read by different serine codon that we know is read by a different tRNA, and now we make wild type levels. So we know it is defective in reading this particular codon in our flag machine. The translation of it is down. So far, so good. However, there are actually dozens of TCA codons in these flagellar genes. Remember 60? This is the only one we saw. And Oki was looking at LAC-Z, and there are several TCA codons, and it was only affecting 2 of the 9 codons, not the other 7. Isn't that weird? I thought it was weird. So that's when we started thinking about context. Is the effect due to context? That is, it's not fitting with neighboring tRNAs or mRNA codons in that flag M. Okay? But in all the others, there were different adjacent tRNAs and adjacent codons in the other genes. So Fabienne did an experiment. We decided, let's just make some silent changes, so-called silent, synonymous changes. Right? Here's that serine codon. Next door is threonine on the 5' side. Proline on the 3' side. So there's 4 possible threonine codons. And there's 4 possible proline codons. So just in a wild type serotype background, so this is just wild type. We're just asking, maybe there's some weird effects of codon pairs, right? And we did an assay. Remember, flag M inhibits sigma 28. And now our readout is luciferase. Okay? So when M is shutting off sigma factor, we're not turning on the lights. Okay? And here's our codons again. Here's serine, here's ACC, UCA, CCU. It's our wild type set in the ligand. And we looked at the effect of changing these codons on luciferase. So if the amount of luciferase goes down tenfold, that says that our flag M activity is tenfold up, right? So I'm flipping the numbers. So this means that it's down 11-fold. So here's our wild type threonines. If just looking at the threonine codons, nothing else changing. Our wild type codon is the worst. Change it to any other, we have 11-fold, 13-fold, 15-fold. What about if we just make the proline changes? Here's our wild type proline. We can keep our threonine 70% up, 20-fold down, just by making synonymous changes. Now, if we take ACU, which is 11-fold up, CCG, which is 20-fold down, and make the combination, what do we get? 34-fold up. Huh? So it's not additive. Somehow they're talking to each other. And we call this the triplet of triplet codons for translation efficiency. So how efficient a particular codon is read depends on its neighboring 2 codons. Okay? And I had this is a great experiment that I had my undergraduate class do. This is the flag M gene. And here's that CCG, right? That is 20-fold down. And we did a tetra. We replaced the first 25 codons with a tetra. We came with an oligo, and we designed that oligo so that every single oligo had a synonymous change in one of those codons, just randomly mixed. And we asked for replacement of the gene, keeping that CCG. what synonymous changes could rescue translation. And we just sequenced at random, and of course we got them all over, which was fine. But it's guys that could suppress it were only the 3, serine, and extraordinary. Again, this triplet codes for efficiency. Okay? I love that. Same as. All right. Now we're going from flagella gene expression just by following this mutant. It's taking us into mRNA translation. Did you all learn this? Once upon a time. Elongation factor bound to GTP when it's translating and recognizing the A site. It goes into that A site 1,000 times a second, just randomly putting in tRNAs. Most of the time it's wrong, right? Pops in, pops out. But if it's close, if the codon is correct or almost correct, it'll stick. It'll stick in this GTP hydrolysis step, which provides it time to differentiate near cognate, things that are close but not right, from the correct codon. And if it's not right, it ejects it again. But if it's correct, it'll keep it, it'll push it over by 3 bases and go again, right? This is just a refresher. Okay. Those of you that hated translation. Okay, so we think that the ability of EFTU to recognize that codon depends on who's in those adjacent sites. And then as he moves down, he's going to influence the next 2 codons. That's our take-home. So far, so good? Okay, now we're going to do some genetics. Are you ready? All right. All right. Is it possible that we can use genetics to test this codon context effect? In general, in vivo mRNA translation. We didn't want any in vitro artifacts, just in vivo. And we needed a system that was insensitive to protein and mRNA stability, right? You can put in different synonymous codons and maybe the mRNA is unstable. Or you can put in different codons for different amino acids, and maybe that protein you're assaying is unstable, right? So people in the 80s, they kind of stopped working on this because it was just too much work. But we're geneticists. Okay, remember this? DNA, RNA, protein, and in bacteria, transcription and translation are coupled. Yeah. Wow. And sure enough, there was a system. And that system was discovered and characterized by my PhD advisor, John Roth. He spent the early part of his career, he studied as an undergraduate with Black Operon, Jacob, Lowe, Repressor, and so he started working on histidine regulation. And when you're starving for histidine, you turn on the genes, right? And so he was looking for the repressor, right? Thinking it was like black eye. No, it wasn't like black eye. It wasn't like black eye. So this is when I joined the lab a long time ago. So I knew this in my head, right? And that's what made me think of using this. All right. Your basic gene, DNA, promoter, or RNA polymerase bond. You have a +1 mRNA start site. You've got a 5' UTR, ribosome binding site before your coding sequence. You've got coding, stop, and then 3' UTR with either a row-dependent or row-independent terminate. The his operon is not your basic gene. Instead of having a 5' untranslated region, it has a 5' translated region. And here it is. So with it, between the promoter and the structural genes, there is the sequence that encodes a little 16 amino acid peptide. It's called the his leader peptide. And there's these stem loop structures, including at the very end, beautiful, very strong row-independent terminator. Knocks off RNA polymerase, just like we talked about. Okay. So it turns out what he discovered, if you have high levels of histRNA, you don't want to make the histone. You don't need it. And so that ribosome starts, and remember, the very first ribosome is fitted to RNA. In fact, Max Gottesman showed the NUS-G protein actually holds them together. Then as soon as it hits the stop codon, polymerase breaks away and keeps going. Okay? And so if it's high his, it reads and it stops over that stop codon, and that protects certain RNA sequences that allows the terminator to form. But they can form alternative secondary structures, such that if you're starving for histRNA, you stall in those 7 his codons in a row, 7 consecutive. And now it doesn't quite hit the B, and so B is free to pair with C, and so C is no longer paired with D. D can pair with E, and now you lose the attenuator. Okay? So the real repressor was actually charged histRNA, and now you can go into the structure. Pretty cool. So this is a system that's independent of protein stability. No one's ever seen that leader peptide. This is an instant. You turn it on, transcribe, translate, boom, you're done. There's no mRNA stability involved. There's no protein stability involved. So that answered our criterion. And the beauty of this system is the default. If you don't translate, you have A to pair with B, C, D. You have the attenuator. So the system demands that the ribosome get on. It demands that the ribosome stalls in those his codons before you're going to express the gene. And this just shows that we can, using lambda red, we can replace the his promoter with TET A promoter. So we can have TET controlled expression. We can add tetracycline. And if we have a stop codon at the his5 position, we know the ribosome is going to stall in those his codons. And it's going to express, this is a RT-PCR mRNA assay. So if you look at the his-G gene, the first gene, we can see that the RNA is being made. But if we inhibit translation, it's not. Again, the ribosome has to get in there. It has to stall in order to read the gene. Okay? So Fabian did a proof of principle. That's where we got that stop codon at his5. She just put stop codons at each of those his codons and asked what happens using a his-D-MUJ, his-D lac reporter. So of course lac is very easy to assay, as we talked about. I hope you've got that figured out by now. His-D is a very difficult enzyme to assay. In fact, all the his enzymes are very difficult to assay. Lac is not. And she found de-repression, and that stop codons at his4 and his5 gave us the best de-repression. Deletion of the attenuator gave us the full level. So we only got half of what we could by deleting the attenuator. We think that simply, maybe there's like a 50-50 chance where you can still form these attenuators. It's okay, still plenty of expression. All right, so now let's do an experiment. Take that his5 codon, and again, using lambda red, put in a tetra, replace it with NN. Yeah. And ask how does that affect de-repression? Again, using the his-D lac reporter. If we're stalling, if translation's slow, it's going to be lac plus. If translation's fast, it's going to be lac minus. Okay. Here is the wild type. We don't need much histidine in the cell. They're pretty white on the copy. If you get rid of the attenuator, we're very lac plus, red. You can see the bile salts precipitating in there. It's very high level expression. Let's do NN and make all 64 constructs. Bit of work. And then assay beta-galactosidase. Okay? Ready? There it is. Beta-galactosidase. Oh my god, who wants to read this slide? Oh, this sucks. Here's our wild type, 11 units. These are stop codons. These are close to 300 units. The better way is to simply take all those beta-gal activities and divide it by the wild type. So we'll look at relative levels compared to wild type. It's a little easier. And I'm showing you their colors on McConkey plates based on the white to pink to red. Right? And again, here's our wild type. This is 1. Stop codons are way up there, 25, 27. And we got some pretty interesting information. For example, look at these protein codons. 1.5 to 3.3. Look at these serine codons. We see variations. So even though these are synonymous codons, they're not silent. So we now see what the mathematicians have shown us, have known since 2000, that synonymous codons are not silent. So different codons, tRNAs are translated at different efficiencies, and we can see that in this assay. We can see other things. These are the rare arginine codons. Fits, they're slow. They're slow. We can see, so remember these balls are individual tRNAs. So these are tRNAs which have a G that can recognize either a U or a C in the third codon. And the biochemists have shown that it goes through a C faster than a U. So if you see it as faster, it would be a lower number. Here it is, lower number, U higher. So we can see this in vivo. Individual changes where a particular tRNA is choosing between U and C. I was expecting, hoping that maybe I'd get like 30% differences, 70% differences, not fold effects, not 5. I mean, it was just beautiful. Now, remember, we're interested in context, right? So what about context effects? So what we did, we put that sear T red codon, because we know that codon is read only by one tRNA, at either 4 or 5, and we randomized either the 3' codon or the 5' codon. So what's the context effect? Context effect, depending on if you're on this side or that side of a specific codon read by a specific tRNA, right? Again, using HIST-LAC assay for effects on deattenuation. And there it is. So I'm showing you here NNN UCA, and below that UCA NNN. Same codons, just different orientations. You look at phenylalanine. If it's phenylalanine first, it's fast. If it's second, it's slow. Then you're tyrosine. So we can see some different effects depending on whether the codons are like this or like that. So we can really see context effects. Of course, the stop codons are always slow. Cool. Cool. All right. Now comes Madison's project. Can we try to figure this out? So back here, this was a really good one. The tyrosine in one orientation was like a stop codon, and here it's super fast. So can we use this to try to figure out what this is? This is genetics. Can we select for faster translation? So here we have our, again, using the HIST fusion. If it's tyrosine serine, it's fast, white. Serine tyrosine is slow, or red on McConkey. Can we make it faster? This is how we try to figure things out. Okay, so Madison set up a selection where she set up 2 copies of the dysregulatory sequence. In one copy, we have our LAC fusion that we've already been working with. But in the other copy, she put in SAC-B. So SAC-B, if it's expressed and you add sucrose, you die. What's that? Death. You guys are getting better and better. What about LAC? We can use this to select for death. This would be a screen, right? So we can select for sucrose. So here we have our mutant, which was the slow one. That's here, anyway, it is fast. It's like this G level of expression, and that is dead on sucrose. Here's the wild type. It grows, right? So we're selecting for soup plus. And of course, you can just knock out SAC B. That's why we have to have 2 copies. So we want SAC B not being expressed, and then LAC. So we can select for soup plus and then screen for those that are left. Okay. Going from a G to an A. There we go. Going from this, here's our selection, here's our screen. What do we get? We got mutants. We got some RNA polymerase mutants in the alpha subunit or the RPOD subunit. Rare, but if you don't transcribe, you're not going to transcribe. If you're defective in transcription, both SAC-B and LAC are going to be down. Right? So you predict that. And then anything general on transcription or general on translation. They're a general translation defect. And so there's RNA polymerase. You can't have translation if you don't have transcription. And we got this base substitution in this 3' tRNA. And we got another base substitution in a leucine tRNA. So that's good. So are these just general translation downs? Here's the 3' mutant. It's in the anticodon stem. And 3' U is essential because it's the only one that reads the ACA codon. And in fact, if we just look at a LAC operon that's not under his control, let me see. This is just a LAC operon unrelated to his, and we can see translation is down if we have 3' U. So it's a general defect in translation. So both SAC and U. What about the other guy? Leucine. Leucine is essential because it's the only tRNA that reads the UUA codon. Okay? And this, way over here, is an A25UG, what's called the D stem. What is going on? So here's our assay again. This is slow, right? And then with Leucine, it now became fast. So we ask, is it a general translation defect? So we have other codon pairs that are very slow, like proline, arginine. They're very slow. So if this is a general translation defect, he should be fast or white here as well. And oops. And the answer is no. So if UCA is there, we see it's down. Okay, it's getting faster. But no effect on the other guy. So it looks to us like it's specific, perhaps, to UCA. And then So the question is, maybe that leucine tRNA can recognize the serine. We're starting to think that, right? And so we had a bunch of UCA pairs that were slow, and so we can ask, is it affecting other things with UCA? And in fact it was. So when we put in the leucine A25G, everything got faster in all those pairs. So it looks like it's actually recognizing the other codon. Then we ask, is it actually putting leucine into that serine codon? And Madison found a really nice paper in yeast where EGFP, there's a leucine to a serine change that turned the lights out. Okay? It was no longer fluorescent and it was unstable. Okay? So we put this in salmonella, and sure enough, no longer fluorescent and it was unstable. So that's pretty cool. So then the question is, if we have our leucine, does that suppress it and put the lights on? And the answer is no. God, life is so hard. You just want an easy answer. This is what we were predicting if it was misincorporating, but it didn't. It stayed dark. Okay? So we decided to push a little harder. This is a mass spec analysis of the protein. Here's our L201S, and if we analyze it at that 201 position, we see serine. And if we have that, Z, we still see serine. Well, we try to help it a little bit. Remember that serine mutant that's kind of defective? You see now, see a little bit. Now, if we combine it with the serine mutant and our A25G, we see 20% leucine proliferation. So it can, but not so well. Yeah, it hit me. In the HIS system, we're always looking at the first ribosome behind polynucleotides. In this system, it might be only one of 200 ribosomes. After that first, you've got the second, you've got the first, you've got all the others. So maybe what we saw in the HIS leader is because we have the first ribosome connected to our polymerase. Okay. And we know it's sensitive because we look at like two prolines behave like a stop codon. And people have characterized this thing called elongation factor P for proline, which helps when the proline comes out, it comes out bent in the ribosome, and this protein gets in there and kind of straightens it out to help the polyprolines. But to see the effect, they had to have like 4 prolines in a row before they saw any effect. We put in 2 in our system, and it's like a stop codon. Again, here they're looking at all the ribosomes. We're just looking at the very first. So we take that very first ribosome in our system that gives us a very large amplification in the translation efficiency. And then here's a paper that came out from Carlos Bustamante's lab. And he actually showed that that first ribosome behind our, it's actually pushing it. It's right on top of it. And it's forcing it to make actually some base changes. So there really is a close connection between that first ribosome. So the guy that discovered the connection, G, between RNA polymerase and the first ribosome, Max Gottesman, I said, Max, how can I make every ribosome look like the first ribosome? And he said, just add a translation elongation inhibitor, that'll allow the effect, give it more time for that LUZ to recognize the codon. And sure enough, what Madison did is we put cells in the plate, added antibiotic, let it grow, you see death, but then you see, she took samples after death, distances from the zone of death, where we still see the antibiotic effects of inhibition of translation, and now we see incorporation, and now we get lights. The GMP is actually incorporating that leucine in that serine codon. This is tetracycline, this is chlorphenicol, this is spectamycin, and kasugamycin is a translation initiation inhibitor, no effect. So everything worked perfect. All right, so that was great. Maybe it's reading the wrong codon. And then I asked 3 people, has anything like this ever been seen before? Darby Hughes, who Stanley mentioned, worked on elongation factor, a friend of ours. Rachel Green at Johns Hopkins, she studies translation in vitro, famous biochemist. And of course, Venki Ramakrishnan, who got the Nobel Prize for the structure of ribosome. And they both sent me back the same answer. Yes, it's been seen before. In 1971, David Hirsch published a paper where a tRNA mutant in tryptophan could read not only the trip codon, but the UGA stop codon. So UGG and UGGA. And it was not in the anti-codon. It was in the D stem. And Rachel told me, yes, the Hirsch suppressor, but she also sent me her paper where she characterized it in a biochemical characterization. And what you can't read, but what it says, is that she found it had an accelerated rate of translation. So that trip tRNA mutant was actually reading codons faster. Well, that was our assay, right? So that was cool. Here it is. It's a G24A, where ours was an A25G, so right, pretty similar. And he originally isolated this that could read not only UGG, but UGA, and then Rachel Green showed it would also read CGG, which is also And then Benke, there you are. Benke sent me his paper. He said Dave Hirsch, and he crystallized it in the ribosome with other tRNAs. That's something I couldn't do. And so this blue is the wild type trip codon, trip tRNA, and in the east found that this tryptenium is bent to this big bend. Okay? And he can show you what happens when it bends. Doesn't matter. So now we hypothesize that our guy must be bent. And it's known that when tRNAs are incorporated, remember that is that slow GTP hydrolysis step where it's near cognate versus cognate. tRNAs undergo a natural bending so that EFP, GTP hydrolysis gives it this time, and then hydrolysis, it says yes, bounces back and moves on. So that's been done by a lot of people. There's a hinge region where this occurs, and guess what? Positions 24 and 25 is in that hinge region. Yeah? So, this is the N, folks. Elongation factor, bring it in tRNAs. It gets to this step, and it undergoes, if it's a near cognate versus cognate, it undergoes this bending reaction, GTP hydrolysis. If it's correct, it stays. If it's incorrect, it comes off. So we hypothesize that we simply bypass this bending step. We already come in pre-bent, and so that allows it to be sloppy. So that's where genetics takes you to places you never dreamed you would go. That's the point of the story. And we never even funded to work on this. I just wanted to figure it out. All right, thanks guys.
We had talked about finishing a little earlier today and having a little bit longer break at the end so that we could just talk to say goodbye to everyone. Right? But I understand people who imagine drank a lot of coffee. So maybe we should take a real short, like 5-minute break, potty break, get some more coffee, and then we'll come back and then we'll have time for our real break at the end, okay?
Okay,
And,
awesome.
and the goal is for us to finish at about 11:30.
¿Listo? Bueno, Professor Marta is already giving us the pray for today. So in case you don't have it, I think she's going to pass on a few copies. So, okay, I'll do it.
Okay, great. Kelly gave a really nice example of what we could call atomic level genetics, where you can use genetics to dissect the finest kinds of structural interactions in really amazing ways, right? But he started also with an example of how understanding certain genetic tools can allow you to build a company that can have real biotechnology value, right? So I'm going to slow down a little bit. I first want to tell you something. Somebody asked me yesterday at the cantina about someone asked how I got into genetics. So I'm going to give you a very short explanation of that, and then I'm going to tell you about one of the companies that I co-founded that uses genetic approaches. There will be a little bit of non-genetics in there to show you that it really does work, right? But that will just give you an idea of some application, all right? Okay, so I was an undergraduate. My older brother and I started a company, and it was going very well. And in my very end of my senior year of college, I took a microbiology class. I said, oh, I love this. This is what I got to do. So I told my brother, well, you know, I'm not going to run the company with you anymore. So we sold the company, and he went off and did something, and I went to graduate school. But it was the very, very end of my undergraduate education, and I hadn't applied for graduate school. And so luckily, I went to a little university nearby, and they said, yeah, we'll take you, come on. And so I started working there, but it was not a very research active place. So I got really interested in this organism called Microcycus flavus. It's a different looking bacterium, huh? When you looked in the microscope, it looked like a little donut. And it's because it grew in these little structures, right? And so I was doing some things, and my advisor said, oh, you should try growing it in MRVP medium. Right? Most people know MRVP. The key thing about MRVP medium is it has a very high phosphate concentration as a buffer, right? But look, this goes in low phosphate for forming little small things, often these little rings, boom, big filaments. Only difference between this and this is the phosphate concentration, right? I said, wow, I'm a beginning master student. I said, cell division is an important problem. And because this is phosphate, and we have ways we can follow phosphate in the cell, I'm going to be able to figure out how cell division works, and I'm going to be famous. So what I did was, I grew bacteria like this, and I grew them like this, under the different conditions, and I added P32, high level of P32, because it's diluted out here, right? So very high concentration of P22. I fractionated all the components. Something weird. I fractionated all the components of the cell. Now, when I was a student, chromatography wasn't what it is today. It was a lot of work. I took these cells, I fractionated proteins, carbohydrate, nucleic acid, lipids, small molecules. I separated everything into fractions, and I compared this and this, right? All of the changes, when I looked at it, the only substantial change was a change in the membrane lipids. There was a decrease in phosphatidylchlorine and an increase in diphosphatidylglycerol. two really important phospholipids in most membranes that are changing under that situation. The reason these guys are long, it doesn't show up very well here, but those filaments are actually cells that are connected, and they have membrane bundles in between each one of these long things. So I did a lot of electron microscopy too. Okay. Did I figure out cell division? No way! I just got another problem, and what else am I going to do? How am I going to solve that problem? Right? So I said, I want to go somewhere where I can learn genetics, where I can now use genetics to ask the question, if I make mutations that do this, will it have that phenotype? Right? So it was because of this thing that I got really interested in doing genetics. And so I left my master's degree and I went on to a PhD program. And I went to work in this guy Bill Nunn's lab. And it ends up that this is me. And after a little bit, this character came and joined the lab. So he was an undergraduate in the lab where I was a grad student.
I look exactly the same, huh?
And then he moved to John Roth's lab. There he is. And a little bit later, I moved to John Roth's lab. So he has a PhD there, and I was a postdoc there. Right? A long time ago. Well, I don't know. Depends on how old you think we are. But since then, we've both really been enthused with this idea of fishing for mutants, right? And the idea that to successfully fish for mutants, you have to really have a lot of plates and a lot of hard work, but you have a lot of fun, right? So that's the story of how I got into genetics and how Kelly and I met. But let's talk about this company. So I was a co-founder of a company that was tied. Initially, we were trying to develop vaccines. It ended up changing to be an anti-cancer company. And the idea here was to use bacterial mini-cells to deliver in a special way key antigens or anti-cancer agents that allow us to do it. The company is called Vaxion Therapeutics because we were starting it as a vaccine company. So here's what I'm going to tell you today, just so you can see it. I want to tell you why, a little bit about current approaches. I'll tell you how many of you know what bacterial mini-cells are? All right, I will tell you what bacterial mini-cells are then. And we'll talk a little bit about some tricks for antigen expression and delivery into eukaryotic cells, how well the thing works, examples, the safety of it, and how we pivoted to anti-cancer. That's where we're going. Okay, there's a lot of current approaches for making vaccines, and each one of these approaches has some good parts and some bad parts. So very often, to find the best vaccine for something, you need to use one thing or another. mRNA vaccines right now are very popular. They're really successful for some kinds of things, but for certain types of things, they're not as effective, right? There are a lot of experimental approaches that people have used for vaccines. And some other people before us that started, Roy Curtis III, if you know that, had started using something similar to what we're doing as an animal vaccine. But here are the key things you have to have. Any vaccine, it's got to be safe. If it's not safe, it won't be fine. Ideally, you want to have at least some response with a single dose. Some people may be able to come back, but some people may get one vaccination and never come back. So you want some productivity there. Ideally, you can give it orally, because a lot of people hate having shots. You want it to elicit both mucosal immunity and peripheral immune responses. Okay? So the mucosal immunity, for example, you secrete antibodies into your gut, and it can prevent the pathogen from even getting into you. But if it escapes that, you want to make sure that you have antibodies in the blood that are able to get it. This response needs to be long-term. You don't have to get vaccines too frequently. You need to be able to make a lot of it. You need to be able to store it really well. If you're going to be shipping vaccine to different places around the world, ideally it doesn't have to have real strong cold storage. So ideally it's stable at room temperature, and in a good case it gives you enough immunity that it promotes herd immunity in your population. All right. So I had been working with some people, and they were trying to overproduce some neuropeptides, and they exist inside the membrane, and they're having a really hard time doing it. So I said, there is this old technique called bacterial mini-cells, and we could probably use it for that. So this is a wild-type bacterium. Wild bacterium, it grows, the DNA multiplies. Ultimately, that DNA separates, the cells separate, and the DNA is segregated into the chromosomes segregate into the two cells, and you get two baby cells just like parent cells. Okay? Now, this process, you don't want it to divide too soon, or you wouldn't be reproducing effectively. So there's a couple of proteins that do this really well. So these two proteins, MinC and MinD, they move back and forth between these edges of the cell, so that this protein, FTSC, that pulls the cells together, it can't form there. Okay? FTSC can't cross through here because the nucleate prevents it from doing so. Right? So these two guys inhibit FTSC from binding there, so that when the nucleide separates, boom, right in the middle, that's where you always get your cut. Now, if you start with a MinCD mutant, what happens is, as this cell grows, FTSC sees this spot here, and you get cell division. You bite off a little tiny piece of a cell, and you get two copies of the chromosome here. This little piece is too tiny, a chromosome will not fit in it. So this is achromosomal, but otherwise it's just like the cell. It has LPS on the surface. Right? It has ribosomes. It can do transcription. It can do translation. All of that machinery exists in that little mini cell.
It doesn't have DNA to do the transcription. Unless you have a plasmid.
So this little cell here, if you have multi-copy plasmids here, the multi-copy plasmids are separated throughout the cell, and they can be in here, right? So a plasmid will fit, but a chromosome won't. Okay? So this is basically the idea. Here's your little plasmids. I anticipated his question. The plasmids can exist in here, and anything in those plasmids you can get transcription, translation, and export outside of the cell. All of those functions. All right, so a pretty cool little machine that allows you to work with plasmid encoded things and eliminate all this other stuff. Right? So, a problem with this, though, is that this process, where you get the MinCD mutant, and this sometimes happens, the frequency is too low, right? That's one problem. And the other thing is, MinCD mutants accumulate secondary mutations at a very high frequency, so it's not stable, right? So we wanted to use this as a factory for producing vaccines. So, so here's the basic things that we thought. We thought, okay, if FTSC induces this sector, instead of worrying about MinCD, let's make more FTSC, right? And the way we make more FTSC is by using a red spot, right? So these are a set of genes involved in cell division. FTSQ, A, and Z. The promoter for Z is located within FTSA. Right? So what we did was we made a little derivative that we stuck in here, and that little derivative makes lac IQ, so we can turn off p-TAC, so we can make low levels of p-TAC when we want it. And it contains the pro-B and A genes. Now, the reason for the pro-B and A genes is because we need to have a selection to bring this into the cell. Right? And if you're making a vaccine strain, you don't want to have tetracycline there. Right? You don't want to have ampicillin. You don't want to have chloramphenicol. You don't want any of those antibiotics because there could be some residual antibiotic that would contaminate things and potentially harm them. So instead, we brought in these genes that are essential for amino acid synthesis, and on the chromosome, we have a mutation. So these guys can't grow on minimal media unless they have this insertion. Okay? So we replace this on the chromosome now. All we have to do, add IVTG, crank up FTSC. Here's the difference. Here's our min-CD mutants. This is the number of mini cells. That's when we turn on FTSC. Okay. Not bad, huh? Now we have a way of very effectively making high levels of mini cells. Right? So we're making 10 to the 12th mini cells per milliliter. All right. So now we have our little factory where we could make something inside these mini cells. But we wanted to think really hard about what we're going to put in there, what our plasmid is going to be, right? And so one of the things we wanted to do was say, can we express protein antigens effectively in the mini cells? As a first test, what we did was we set up a system so we're using the Ramnos promoter. And this promoter, some promoters are on really lots or off. The Ramnos promoter you can turn on at very different levels. So it's a rheostat. So this promoter, we just in this case are hooking up a GFP protein behind it so we can look at it. All right? So let's see what happens in mini cells when we turn this on. This is before we've induced mini cells. We haven't turned on FTSC. This is when we give a short turning on of FTSC. And this is after longer induction, but you can see the little mini cell budding off. And this is our purified mini cells. We can very effectively produce that protein inside the mini cells. All right. We wanted a vaccine that would give us very high levels of immunity. We didn't want to just have a protein antigen vaccine. So to do that, we really would like to see our bacterial mini cells engulfed by antigen-presenting cells in a host so that they would generate a response inside the antigen-presenting cells. The way we figured this out is, okay, this is our plasmid. I've done this before. Here we brought in a eukaryotic promoter. It's not turned on in bacteria, only when it's inside of eukaryotic cells. And down from it is we have green lantern protein. So if the mini cell is engulfed by an antigen-presenting cell and it can express this protein, we'll be able to see that inside of our cells. Go. This is simply light microscopy. This is fluorescence. This is our empty vector without the induction of the promoter. And this is our mini cells inside. These are bone marrow-derived dendritic cells. It works in a variety of ways. So, all right. So what we've done now is we've built a little tool that works in bacteria and it works in eukaryotes. Right? All of this using bacterial genetics. Right? So now we can say, all right, does it really work? Right? We had fun making these strains, but do they really work? All right. So I want to back up for a second. So if we simply make the protein in mini cells so that you're exposed to those proteins, that stimulates this MHC2 class immunity, right? And that generates antibody. If antigens are expressed inside of those presenting cells, it generates an MHC1 kind of response, and that gives you cell-mediated immunity. And, oh, by the way, most vaccines are based on this, or they're based on this, one or the other. But if you do both of these things, you generate both antibodies and cell-mediated immunity. So we're going to look at all of those things. All right. The mini cells can deliver both of these types of antigens. This is an antibody titer here. We're giving 3 doses. No doses. This is a set of these guys who were printed up. And you see we get a really strong antibody response. We've also tried delivering this intranasally and orally. Remember I said that's good for a vaccine. And you can see this is 1 dose given as an injection, high response. This is 3 doses given intranasally or 3 doses given by mouth. This is still a pretty good response. This is enough. If we look at salmonella antigens, this is enough to protect salmonella, protect mice from salmonella. And then we can look at the response that we get. So this is antibodies in the serum. This is a secretory, an IgA immune response, if we give it nasally or if we give it orally. Okay? So we get all of those kinds of responses. This is a plot of mini cells inducing antigen-specific T cells, right? So this is cell sorting. The key thing is that we get this is an anti-interferon gamma response as we go this direction. And the key thing is we're stimulating CD8 T cell response. I don't know how many of you are immunologists, so I'm not going to go into details about the response. We just get it. This is a response showing that you get a really strong cytolytic T cell response. This is a cytolytic T cell. This is a virus that stimulates a strong cytolytic T cell response, and you can see that when we have our mini cells, we generate a strong response. So here's the take home. Using these mini cells, we generate real strong humoral and T cell mediated response. These responses are synergistic. This is synergistic. If we only stimulate one, we get a low level of protection. If we stimulate both together, we get very high amplified expression. A single response generates immunity, and mucosal delivery will give us a very nice secretory antigen response. So now I just showed you a bunch of responses of cells. How do you know that it really works, right? So to do this, we tested our mice that we treated with or without immunity, and we challenged the mice with a nasty viral pathogen. That it's a lethal mouse model. So in this case, we use LCMV, injected directly into the brain of the mouse. It's 100% lethal, a very strong test. Immunity, in this case, requires a strong T-cell activation and cytolytic T-cell activation too. It requires both, because if you don't stop that virus right away, bang, the mouse is dead. Right? This is a control, yes, and the virus. This is mini cells only. They die. In 10 days, all of the mice are dead. This is if we give our mini cell vaccine, mice live to old age. Right? So that's a real strong model for this process. So if you look at this, it's done all of those things that we wanted by doing some simple bacterial genetics. It confers protective immunity, it gives us all of those responses. The delivery system is very easy. The mini cells, we can spin them down on a pellet. That pellet lasts for a long time. The mini cells, because they're not actively growing, they don't die. So we can store them for a long time at room temperature. A single dose is effective, and we don't have to worry about reversion. There's no chromosome to revert, right? So you can ask about safety. And so we made a number of improvements. We made genetic changes that changed the LPS on the surface, so it's not so toxic. We've done all of this so that we have no antibiotics in the host. All of our complementation is complementation of auxotrophic mutations. Right? There's only one problem here. Our mini cells work really good. I told you we can purify them and everything. What do you think the problem might be?
The LPS?
So we've modified the LPS. The LPS that's there, because it's weak immunogen, but it's a great adjuvant. So in fact, it's not enough to stimulate a negative response of the host, but it ensures that this thing works. We don't have to add mercury or something. Okay. What's it? Here we are. Let's just see. Can't see that. Yeah, where's the
Not that Joe?
Did you steal it?
Oh, no, it's okay. It's the black thing.
Okay. Here's our cell. We induce it. Here's what we have left behind. Okay? Here's our mini cell. So then we purify these. And we use this for our immunization. At the scale I've shown you here, no problem. But if we're going to scale this up really high, what do you think the problem is? Even if a very low level of this parent bacterium contaminates the mini cells, we are injecting people with bacteria. That's not so good, is it? Right? So bacterial genetics comes to the rescue. All right. Okay. Remember when I was talking about RRN operands in ICU and it cuts them? ICU is like a restriction enzyme that recognizes a very long sequence. And that sequence is in every single RRN operand. Okay? So I said, hey, any of these guys that are left behind, we want to kill them. And if we cut them in 7 pieces, the chromosome will be rapidly degraded. Right? Moreover, if you have a low level of these and use some mechanism that lyses them, now that releases active LPS that could cause a toxic response in humans. But if we simply destroy the chromosome, they're dead, they're gone. So I said this could be a really good suicide mechanism. Any whole cells that squeak through, we're going to turn on this enzyme in the cell. It's going to cut. Right? And that chromosome is destroyed. Here's the answer. We induce ICU1. These are purified preps. Okay? Here are the, so this is time, and this is the amount of each component we see. Mini cells. When we turn on the system, we get a lot of mini cells there. When we don't have ICEU1, we see that there is this low level of parental cells that sneak through. When we turn on ICEU1, parental cells are on. Okay. By the way, I have to say, we could have purified these so good that we wouldn't have to worry about that. But it would cost a lot of money. By building this system into the bacteria, we're getting rid of those parental cells, and we can do this whole thing on a large scale for really cheap. Okay. It's a series of genetic tools, just like the kinds of things we've talked about in this course. Okay, wait a second. Anyway, here's the problem. We were doing this in 2015, before COVID. And you know what? When you make a vaccine, no matter the best vaccine in the world, there are always some people that have a negative reaction. So companies that sell vaccines have to have a lot of insurance, right? And it ended up that the insurance cost was too much for our little company, right? And no big company wanted to invest in vaccines. They said, we have all the vaccines we need, why are we going to invest in vaccines? So we built we worked really hard to make a nice way of building vaccines. Nobody wanted it, right? Bummer. So we said, well, what else can we do, right? We just did a good time experiment. We said, okay, is there a way of using these mini cells for something else, right? So we built a way of putting in fluorescent probes that target different places in the body. We thought that would be good for diagnostics and imaging. Never caught on. We thought it would work for delivery of inhibitory RNAs. We never pushed that very hard. But we then decided, well, we can load these mini cells with anti-cancer agents and get targeted chemotherapy. Right? The idea here was to get secondary chemotherapy. So there's a lot of chemotherapy for cancers that work really well, and then there's some mutation in the host, and you start to die. Right? So we could have a completely different way of treating those chemotherapy-resistant people. So we basically got that thing out. We developed that approach. We proved it was safe in mice. We took it to clinical trials in humans. It is now in phase 3 clinical trials with a couple of different types of cancer. Phase 3 clinical trials. Phase 1 proves safety. Phase 2 proves that it works on a limited number of people. And phase 3 has a very large number of people. Because we're looking at specifically people who have problems with response to chemotherapy, they're smaller populations. So the clinical trials take longer. We're in phase 3 clinical trials. In those clinical trials, we have already saved people's lives. And if this moves out and gets into the public sphere, then potentially we have a way of saving people's lives from kinds of cancer that there exists no other treatment for. Right? So, and again, I want to emphasize 2 things. One is, this is really based on bacterial genetics. And the other part of it is, this is something that Kelly talked about with the Golden Goose Awards, right? I have got money from the federal government for for many years to do basic science, right? To understand how bacteria use proline as a nitrogen source, to understand a lot of things. And when you think about who's giving you that money, it's the citizens. It's the people who are working in the shops. It's the people who are making us food. They trust us by supporting us with tax dollars. I really appreciate being able to do that, and the idea that I can give back to society is amazingly enriching. All right. That's my story. That's one of my companies. That's a company that's still in business right now. I have to say, by the way, I was involved a lot early on for genetic tools. When it gets to clinical trials, physicians deal with it. I can't offer them any help. And so, right now, I'm just sitting and watching what's happening. Look, we talked about a bunch of things in this course. Let's just quickly summarize those things. Okay? These are some core concepts of bacterial genetics. One, large numbers, right? If you're going to find those rare things, if you're going to find the things that might require two changes, you need to have large numbers. Okay? That is, you need to search through large numbers, and even if when searching through large numbers you only get one, you still learned a lot. Okay? One, it's really important to pay attention to the frequency that you get. Right? If you think that you're going to find mud insertions that are only going to be turned on in one of 6 of the mutants, and you find it's on in everything, there's something wrong. If you think it should be turned on in 1 in 6, and it's not turned on in 150, there is something wrong. Right? So paying attention to those numbers is really important. The strain background is really very important. This is why we talked about back crosses. Those of you who were in the lab will also realize that's why we worry about persistent phage infections. Because if you have something like that, you're no longer able to compare 2 different strains. A selection is worth 1,000 screens. Kelly just emphasized that again today. The pencil is the wrench of the chromosome mechanic, right? So sometimes just sitting and drawing out what you might get can help you make predictions of how to design your experiment. Transposons can give you this tool of portable regions of homology. So instead of just relying on the homologies that God gave you, it allows you to play God. The value of reporter fusions that have selectable and screenable markers. MUD-J, MUD-K, T-info-A, T-pop, and others. When you want a defined mutation, very specific mutation, the ability to make those mutations using red swaps and CRISPR change the way we can do things. Just to focus on this for a second, though, this allows you to construct something you want to construct. But these things allow you to find something that you don't have no clue what it is, right? So both are really important. Once you understand, then you may want to go in and look at it more detail. And finally, what's the final one, Kelly?
You can't always pick what you want.
So that's those are really key core concepts that I think that we hope you take away from this class. If you would like to read more, I'm going to have a little advertising. Kelly and I were editors of this book, a tribute to John Roth. A lot of fun stories about how people use different approaches to understand genetic problems. These are stories. This isn't a textbook, right? But it helps you have insight into the way people think. This is a book I wrote with Roberto Coulter. It talks about microbes and evolution. So it talks a little bit about some of those changes that happen out in nature. Again, these are stories. There's some real fun ones in there. One of those stories is by... how many of you know what roller derby is? No? No? This is a book on One Health. It was I was an early proponent of the idea that if you think about infectious diseases, you can't just think about humans, you have to think about humans, animals, and the environment. This book deals with that and a lot of good examples of it. And if you just want to know everything there is to know about genetics, Kelly and I edited a 7-volume encyclopedia of genetics. And now the final plug is coming out soon. A book that I wrote with a couple of co-authors on scientific integrity and responsible conduct of research. In this course, we have mentioned a couple of examples where somebody's data wasn't right, confused the field. This book is really meant to help us all be better, do good science, honest science, that will really move science forward. Gracias.
Okay, so just to finish the course, I want to show a couple of slideshows. It's not going to work. Okay, it's a little bit too big. Anyway, it's fine. So, first, well, I'm not going to say goodbye yet, but anyway, this is a brief description of what we took Stan and Kelly during these days. So, when we just arrived, we went for tacos really nearby. Luckily for Kelly, Stan had been here one day, so we went for tacos, pastor tacos. So they really tested the real pastor tacos, not the Los Angeles tacos. Not a good imitation, I should say. I mean, they do their best with what they have. But here, the real. Of course, I brought them to the school to take a look at how everything was going. And then Kelly's already trying to do the well-done, so he's going to do his best today. Later, we took them, of course, for visiting Templo Mayor. We went around Templo Mayor, and we went to a very nice, beautiful building. In case you guys don't know it, it's CEP, el edificio de la CEP, which is not open now, and it's almost empty, almost nobody knows. It's for free. You can see a lot of Diego Rivera's murals and also Orozco and every other thing. So I took several pictures. They're going to have all the files. You guys haven't been in Palacio Nacional. Before it was easier, but it's impossible to get in for free again. So we took them to see all the Diego Rivera murals and the Palacio Nacional. Later, we took them to downtown. Well, we visited Gran Hotel de Mexico, and then also, of course, the Museum of Anthropology, which is beautiful. I always never get tired to go and visit that place. It's always nice to see all those, all the history that Mexico has. Is very amazing. So, and then one thing that when Stan was preparing to come, he told me, "Can we go and see that mural that Diego painted for the underwater in Cárcamo?" He didn't remember the name, but I knew about that. So believe me, I was like your age and I never knew about that place. And then in 2010, I think, they reopened the place. They did a really good job. So there's only a few people who know about Cárcamo de Dolores. And the Cárcamo, it's in Chapultepec, second section. So you get there, you get rent there really nice. So you can now do a really nice tour. You can go to Los Pinos and then take the cable bus. It's really beautiful ride. Go on the weekends because on holiday. And then really nearby, you just walk a little bit and then you get to El Cárcamo de Dolores. So inside, I don't have a picture, but you need to look for it. It's just a beautiful building, a lot of history. And then,
Can I add one thing?
Sure, go
If you
ahead.
haven't been there, Diego had learned about microbes and evolution of life, and so it shows microbes and it shows evolution up to people.
Yeah.
If you're a microbiologist, it's cool.
It's a
You
must.
gotta go.
And yeah, and if you're students or professors, then you get a discount. It's not for free, but you get a discount. It's very, very accessible. So then after that, when in our visit for Palacio Nacional, they learned about the magueys. We saw a lot of magueys, which is different than the agave because magueys are bigger. And which beverage comes from a maguey? Pulque. So they don't know what pulque was. They had a natural one and it's Sakurado, the mango. I think all of us love this one. Because Kelly was one, La Malquerida, which is open now again, because you guys. So anyway, and then all knew about this strike, which we know it's a first strike, but it affected a little bit the course among this. But because we went early after the lab course, we were able to take them to Monumento a la Revolución, so we went to the underground and also to go the stairs. Of course, outside, there was a weird guy taking pictures of me. But he was really nice, so he took a picture of us there. And then, Kelly's, both there, you have seen they are so hardworking, workaholics, I think, even when you say you are not. This is a proof that you are. So I saw him yesterday working on the computer on the floor. So for all of those that couldn't be in the lab, it was a fun part. I wish I could have more opportunities to have you all, but limited resources were limited, and then the space and then we wanted them to be in front of everybody. So I'm sorry if you couldn't get a spot there. But then we have a few pictures that Martin took. So I'm not going to say the story of this one. Laboratories are laboratories, so sometimes things work, sometimes they didn't, sometimes they get close. That's one thing. So there were a few there's a lot of pictures, a lot of them. So I have them, they're going to be shared with everybody. So the love for science you see up there, Neil's asking for. So this is a picture that we took at the middle. So I think it's a very good job that he did. So and just can you turn on the lights, please? Well, just a few last things. So just to give the final acknowledgement. So first, to Stan and Kelly, so we have them up. This is just another piece of paper for you guys, but it's just to tell you which color you want, Stan. That's first.
Green.
Green? Sorry.
Irish.
So this is a small present for him, and then this one's for Stan. Hope you guys like it.
¡Bravo!
that. So also, I want to thank, of course, the school because despite whatever happens, the school is always generous. So they let us use the facilities, the laboratories, and all that. So basically, the head of the department allow us to use one of the labs, and then the auditorium, and also the people at the cocina. They also help us to prepare a few of the plates that we use. Of course, people at the cómputo, Ángel, muchas gracias. Carlos, who is the head of that department, Jesus also, and also the people from the Servicios Estudiantiles, despite the fact that they didn't want to use the microphone, but they anyway, I think the videos are okay. Also, thank you to the donators of the snacks and the food during this time. I mean, Eduardo, Abigail, Abby, thank you very much, and the rest that I'm maybe forgetting, the coffee by Margarita, and also, well, the photographer, Martin, who came, and I think he did a great job. And then I also want to thank our students that prepared the plates. So you guys want to stand up? So Jose, I see you there, Wells, Amanda, Orlando, Mario, Jenny. Not all my students are here, but they also helped, despite the fact that we're in the course. So also Nestor, Enrique, this Israel, these three guys are starting their social service in the lab, so they already know it's a hard place to be. Ruby, and well, yeah, thank you very much. And well, that's it.
Can I do one more, gracias?
Sure.
This is from both of us. We really have appreciated all of you as an audience. We really learned from your questions. We liked the way you interact, and we could see whether you were asleep or not. And the thing that was really nice was you weren't asleep. You were all paying attention. So it really is nice. You know, a teacher learns as much from the students as the students learn from the teacher. We felt like we've learned a lot from all of you. So muchas gracias. And then one more person that we didn't thank was Paulina for all of the things, all of the putting up with us and dragging us around town. We appreciate that so much.
Yeah, that was my mistake because Paulina, Marta, they also were a lot of help. Thank you very much. And, one more thing to do, which is the teapot. So my proposal is that we do the teapot song and then Abuelo. Is that okay? Yes. I send you the lyrics for the Abuelo in your phones. ¿Nos ayudas a encender la luz? Gracias. So, I propose that we do it, whoever who wants to be here, and then in the halls and in their seats, so we can spread the whole auditory. Nos podemos distribuir en todo el auditorio y subir los que se quieran subir aquí, porque hay unas 3 filas, no van a saber caer.
Seleccionamos las cosas.
Also, Diana's brought her ukulele, so she says she wants to play together with the song. Is that okay?
¡Ay, esos jóvenes!
some sandwiches, and then to say our goodbyes. We are leaving around 12:30, 12:40 to get them to the airport. Okay, so
Yeah.
this is your opportunity. You wanna get like pictures of them, so
Right.
yeah.
Yeah,